Hello, my name is Jory Van Cleve. Today we're going to be talking about cell cycle regulation, which is a type of signal transduction. Before we begin talking about how the cell cycle is regulated, we need to go over the cell cycle itself. The cell cycle consists of four main phases. The first three phases make up a phase called interphase. The majority of a cell's life is spent in interphase, where it continues to grow and carry on everyday processes, along with replicating and synthesizing DNA. Notice on the image the interphase is noted by the yellow color. The first three phases, growth 1, synthesis, and growth 2, make up interphase. An important factor of successful cell division is ensuring that the cells remain identical. In order for a species to survive, it must maintain integrity of its cells and the genetic information found within the DNA. Mutations in genetic material can be lethal, therefore, many organisms die because of it. Cell growth is essential to the cell cycle, which is what the interphase functions to do. When interphase is completed, the cell will have twice the amount of DNA, centrioles are replicated, and the cell has grown enough to be able to divide. So when you look over here, these three, G1, S, and G2, this is all of your interphase. And again, this is where the majority of a cell's life cycle is taken. And then you get over here to mitosis or meiosis. Within that, there is 4 more stages if it's mitosis, or 8 if there is meiosis. And then the very last part is cytokinesis, where the cell actually splits into 2. The next phase of the cell cycle is called M phase, which includes one of 2 different processes. Again, mitosis or meiosis. Mitosis is the division of somatic cells, whereas meiosis is the division of sex cells. Mitosis will go through a successive splitting where one cell becomes 2 new cells, and meiosis or meiosis will go through division 2 times, therefore one cell becomes 4. Meiosis, meiosis, whichever way you prefer to say it. This is of your sex cells. You have this one cell that goes through your prophase 1, metaphase, anaphase, telophase 1, and then it goes through prophase, metaphase, anaphase, and telophase 2. So this one cell becomes 4 at the end. In mitosis, it's going to go through PMAT 1 time, prophase, metaphase, anaphase, and telophase, where one cell becomes 2 at the end of that. So again, the cell cycle includes 4 main phases. However, notice on this graphic, you have a G0 phase. During this phase, nothing is really going on. It's said to be at rest. During this phase, the cell's machinery and regulators are dismantled and broken apart. This includes the disappearance of cyclins and cyclin-dependent kinases, or CDKs. The other phases include G1, S, G2, and mitosis. Cytokinesis is the end result of mitosis, where the cell will finally split into 2 different cells. The length of the cell cycle is dependent on the cell type. Some cells, such as the cells that make up the lining of our intestines, complete the cell cycle in as little as like 7 to 9 hours. However, some human cells can take up to 24 hours to complete, like a skin cell, or even 60 years to complete, like nerve cells, which is why when there is nerve tissue damage, like brain and spinal cord damage, you very rarely can get back from that because it takes so long to make new tissue like that, new nerve cells. When you go into this, and then sometimes a cell will be kicked back to the G0 when some things are going a little bit wrong and it's got to go back and it makes it rest and start over. So here you have your G1. Some people call it growth 1, some call it gap 1, your synthesis, your growth 2 or gap 2, your M phase, which includes the other phases within it, and then cytokinesis. So this first stage of interphase, G1 or growth phase or gap 1, um, the cell is just continuing to grow. It's getting bigger. While it's growing, it's making and synthesizing proteins. It's also making organelles that are needed to make DNA. During this phase, the centromere and other parts of the centrosomes are made. The cells carry out everyday functions as they just wait for the next phase, which is where DNA is going to be copied. Sliding into S phase or synthesis phase, the cell will make a complete copy of its DNA. Along with copying DNA, the cell also duplicates the centrosomes that are responsible for organizing microtubules. Centrosomes are connected to spindle fibers that are then responsible for pulling those chromosomes apart and moving them across the cell during mitosis. Transitioning from the G1 to the S phase involves the development of replication forks because DNA has to unwind and unzip with the help of helicase. And then those replication forks keep those old strands and new strands apart and keeps the old strand apart when it's splitting. The single-stranded DNA needs to be stabilized with the forks, and polymerases will then be added that will proofread and zip the DNA back up. All of these processes include cyclin-dependent kinases, CDKs, and DBF-dependent kinases, DDKs. And here you can see the old strand that splits apart. So right here is where you would find replication forks. Polymerase is going to unzip that. As the new strands come along and pair up to the correct bases, polymerase will proofread that, make sure that those are all being copied correctly, and then glue those back together. Of course, we all know that it's not foolproof, that there are going to be mistakes and there are going to be mutations to where things slip by. After the S phase, you go into the G2 or second growth phase, also known as gap 2. This happens after the synthesis phase. G2 has two vital functions: to get everything ready for division and to correct any malfunctions or deficiencies that might have occurred. During this phase of mitosis, the cell continues to grow larger and larger. It increases the amount of cytoplasm. It starts making and assembling organelles, which doubles the amount of organelles so that there's enough for two separate cells, because remember, at the end of cell division, that one cell is going to become two, or if it's meiosis, one becomes four. And so it has to make sure that it has enough of that cytoplasm to support two cells and organelles. Mitochondria and lysosomes contain their own DNA, therefore they divide independently, and they do this at the beginning of G2. A vast amount of ribosomes are also needed in order to support two complete cells. The cell also prepares for the nucleus to divide in this stage of the cell cycle. As the cell leaves the G2 phase, proteins that promote mitotic functions are released, and this is going to include protein complexes called MPF, or mitosis promoting factors. And as you notice in this graphic, it just shows the arrows showing to the gap 2, shows this just getting bigger. It's got to get larger so that this can end up splitting into two. By the end of this, it should have duplications of everything in it so that when it starts going through mitosis, it has enough to split. Okay, M phase is the last phase of the cell cycle. This is called the mitotic phase. This divides the cell so that two new cells are formed. There are two parts to M phase: mitosis and then the very last portion called cytokinesis. Mitosis includes four main stages: prophase, metaphase, anaphase, and telophase. During prophase, chromatin begins condensing into chromosomes. The nucleus also begins to disassemble and disappear, and the spindle fibers begin to form. Microtubules are responsible for organizing and arranging chromosomes along with forming the spindle fibers. During prophase, the centrosomes begin to move in opposite directions. The second phase of mitosis is metaphase. During this phase, during metaphase, the chromosomes begin to be pushed by the spindle fibers into the middle. They line up in the middle. They do this to ensure that sister chromatids divide equally. And of course, once again, it's not foolproof. Things can happen and those sister chromatids don't divide equally. And so then that brings on a whole array of different disorders and diseases. During the third stage is anaphase. This is where the sister chromatids are going to be pulled apart by the spindle fibers. So remember that they were lined up in the middle during metaphase. Now the spindle fibers are actually going to pull those chromosomes apart, and they are going to end up moving to opposite sides of the cell, or what we call opposite poles. During the last stage of mitosis, telophase, this is where the cell is almost two complete cells. The cell is elongated, it's got two complete nuclei, spindle fibers detach and start breaking down. It's got its own cytoplasm for both parts, it's got organelles, and all it needs to do is end up splitting down that middle. And it could be the belt of protein fibers if you're talking about an animal cell, or a cell plate if you're talking about a plant cell. But that is the point to where when you get to cytokinesis, that the cell is actually going to split into two identical cells. When you're talking about mitosis, this is going to be your two identical cell splits. And you can see on this picture here, it just shows the different stages. You can see the chromosomes lining up. Well, here you can see where the nucleus is starting to disassemble, break apart. Your chromosomes are now free. The spindle fibers are formed in metaphase, or lining up in the middle. The chromosomes are. Here the spindle fibers are pulling apart the chromosomes. And then you begin to have an elongated cell, which then looks like almost two complete cells, and then it just needs to divide right here. And all of these processes are pushed along with different types of proteins and regulators that help ensure that the processes are taking place as much as they can. So during that cell cycle, you've got things called checkpoints. And these checkpoints are to check each part of that cell cycle, or different portions of the cell cycle, to make sure that things are going okay. So during the cell cycle, the cell has to maintain homeostasis as it's growing. Cell division, cell differentiation, and cell death are all processes that help to regulate homeostasis in a cell. And remember that homeostasis is a stable internal environment. So they've got to stay stable. They've got to be able to do their jobs. The cell cycle checkpoints are one way to ensure that homeostasis is happening, for at least most part that it does. If a checkpoint identifies a problem with the cell, it will first attempt to fix the problem. So for example, it may try to repair the DNA. However, if the problem cannot be fixed, the cell will go through a process called cell death or cell suicide, which we'll talk about more later. But this is when a process called apoptosis occurs, in which the cell self-destructs in order to protect the organism. Uncontrolled cell death, or uncontrolled cell growth, can result from the loss of genetic integrity or the lack of cell death. Uncontrolled cell growth results in cancer and other pathological diseases. The checkpoints carry out various assessments to ensure that cells are not created that aren't functioning or working properly. And of course, like we said, it's not foolproof. It's not 100% effective, but for the most part, it's effective and it catches those cells that aren't working properly. One assessment includes checking for DNA damage. If certain proteins are present at the sites of broken DNA, then the cell will not divide. It'll just cut off division. The checkpoints will also check DNA replication. If the entire strand of DNA is not copied, then the cell will quit dividing, or it's supposed to quit dividing. The checkpoints also check the cell condition to make sure that cell proteins, organelles, and other structures are in high enough quantities to support 2 cells. One of the last things a checkpoint checks is for cell stress. If the cell is under stress, cell growth will stop. I've always learned, and everything I've always read and researched showed that there were checkpoints in 3 main places. One was after growth 1, one was after growth 2, and one was after M phase right before cytokinesis. This particular graphic that I found showed that there was one actually at the S phase. So I thought it was kind of interesting, so I thought I would include it. So the first one was at the cell growth checkpoint in G1. It said it happens toward the end of it. It's just going to check to see if it's big enough, to see if it has the correct proteins. And if not, then it will kick it back to, remember what I was talking about earlier, it'll kick it back to that G0 phase so that it's resting and kind of get it re-kicked and restarted. This one says that there's a checkpoint after synthesis. So this is going to happen during synthesis. It's going to make sure that the DNA was replicated correctly, and if so, it's going to allow it to go into M phase. I've always been taught that that was at the end of G2, so I thought it was interesting it was here. Even when it's at the end of G2, it's also checking to make sure that the DNA was synthesized and copied correctly there. So that may be why it's both showing that way. And then the last one is right after M phase, before it goes to cytokinesis. It's going to check and make sure that all the chromosomes are attached, they're all lined up correctly, that sister chromatids are together, before it allows it to go through cytokinesis to actually split into 2. So cell cycle regulators, cyclins, cyclin-dependent kinases, and inhibitors all help to regulate the cell cycle. So it's regulated and pushed forward with the help of those proteins. These proteins are responsible for movement through the cell cycle as each phase is reached. The cell cycle is also regulated by 2 different types of inhibitors, including the INK4 and the CYP-KIP family. The INK4 proteins include the P16 and P15, whereas the CYP-KIP family includes P21, P27, and P57. The inhibitors function to block certain cyclin CDK complexes, which then stop the cell cycle at specific points. When you look over here at this regulation of the cell cycle, you can just see how the cyclins and the CDKs work together. They help with the regulation, and it just shows at which points of the cell cycle those are found and kind of what their jobs are. So there are 4 main or major classes of cyclins, A, B, C, and D. Cyclin B is found in the cytoplasm of cells. Cyclins A, D, and E are all found in the nucleus of cells. There's 2 types of A cyclins, A1 and A2. When there's a deletion of A2, when that occurs, it is lethal to the organism. A2 is essential to the early embryonic period and cannot be compensated by other cyclins. So it's vital. It has to be there when the embryo is developing. There are times, however, when A1 can compensate for A2 during early implantation, but just not the early embryonic stage. But during implantation, it can compensate for it. There are 3 different B cyclins, B1, B2, and B3. B cyclins are a vital part of mitosis. B1 and B2 make up the majority of cyclins in cell division. Cyclin B1 helps in the development of microtubules, and B2 is associated with intracellular membranes. Even though both play big parts in cell division, they both have completely different roles in the process. B1 and B2 work hand in hand with CDKs 1 and 2 by means of breaking down the nuclear membrane, condensing chromosomes, and developing spindle fibers, all of which are important steps in cell division. B3 is found mainly in the nucleus and works with CDK2. Cyclin Ds are proteins that are short-lived. They, along with CDK4, are dependent of mitogenic signaling in the growth 1 phase. This protein is able to cycle through each phase as long as they continue to receive stimulation from the mitogenic signaling. Cyclin E is a protein that is most profound and found mostly at the growth 1 and S phases, with cyclin A increasing at the S phase. Each cyclin continues to interact with CDK2 and CDK1 as the cell cycle progresses. You notice on this chart here, it just shows how cyclin D is kind of found kind of equally throughout the cell cycle. It kind of goes up, kind of peaks around S and G2 and goes back down. Cyclin E, as you notice, kind of begins at G1, peaks right at the S phase, and then comes down. Cyclin A starts increasing. It peaks at G2 and then decreases. And then cyclin B increases, peaks right there at the beginning of mitosis, and then goes down. So this just kind of gives you a visual of the concentration of each of those cyclins at each phase of mitosis. Okay, cyclin-dependent kinases, or CDKs, are protein kinases that play a huge role in the regulation of the cell cycle. These proteins only get activated when they bind to cyclins. The human genome consists of 21 genes that encode CDKs, which includes 11 classic CDKs. They're numbered 1 through 11. There are 3 cell cycle-related subfamilies, CDK1, 4, and 5. CDK4 and 6 promote the start of the cell cycle. Each CDK is paired up with a cyclin, and all CDKs are consistent in quantities throughout the entire cell cycle. Each type of cyclin CDK complexes has the task of modifying groups of protein substrates. Phosphorylation of the substrates has to happen at just the right moments so that the cell cycle can continue to progress. The life cycle of a cell is dependent on the processes and events carried out by these enzymes and proteins. Cyclins and CDKs are charged with the most important task in the cell cycle regulation. The cell cycle can be sped up, slowed down, or even halted, dependent on the presence and action of those 2 types of proteins. Dysregulation of the cell cycle can cause major diseases and disorders. Cancer, cardiovascular disease, infections, inflammation, neurodegenerative diseases can result from the cell cycle being compromised. Uncontrolled cell growth is the result of impaired regulation of cell death when bad cells continue to make it through the checkpoints. Uncontrolled cell division can occur because of dysregulation of the cell cycle. Genomic instability can cause multiple mutations, which then affect cell death regulators. Dysregulation of the cell cycle and cell death are both physiological processes that are necessary for autoimmunity. This just shows a normal stem cell. It differentiates, makes tissue homeostasis. But then, if there's a cancerous cell, uncontrolled cell division, it starts making that tumor cell and tumor growth, and it just has rapid uncontrolled cell growth. As the cell progresses through the cell cycle and passes through each checkpoint, it is evaluated to see if it's healthy and if it has all of its parts. If it is missing something or if something is mutated, the cell then attempts to make repairs. If repairs cannot be made, the cell will then basically commit suicide by means of apoptosis, necrosis, or autophagy. There's actually several more types of cell death, as you can see on the graphic. Apoptosis is also known as programmed cell death and is considered the fastest route of cell death. A proteolytic cascade is formed that induces the apoptotic pathway, which causes irreversible cell death. There are 2 pathways that are included in apoptosis, intrinsic and extrinsic. The extrinsic pathway is known as the cell death receptor, where death ligands bind to stimulate cell death. Intrinsic pathways are known as mitochondrial pathways. and they create a megapore which covers the inner and outer membranes of the mitochondria. These lead to cell dismantling. The third type, necrosis, includes many routes of cell death. This type of cell death occurs when there's an extensive damage that leads to cell integrity destruction. This can be high temperature, mechanical stress. The passive process does not require any type of stimulation, but it can result from a signaling cascade. Autophagy is another type of cell death that is caused by nutrient deprivation, hypoxia, oxidative stress, genotoxic stress, and high glucose levels. When this happens, vesicles in the cell are formed that engulf the cytoplasmic structures. This process is also regulated by various types of proteins. So dysregulation of cell cycle or disruption of cell death can cause many different types of diseases and disorders. Some things such as cancer, neurodegenerative diseases, premature aging, and cardiovascular diseases can occur. Most people just identify checkpoints and dysregulation checkpoints with cancer because it can cause uncontrolled cell growth, which is what causes cancer. But there are many other things, as you can see on the chart, that can happen, such as viral infections, autoimmune disorders, blood disorders, metabolic disorders. All of those can be brought on by disruptions in the cell cycle. So even though most people don't realize it, and we don't think of what exactly causes those, many of those things are just from disruptions of the cell cycle, which can be caused by an array of things like environmental factors and other things that disrupt that. Anyway, that is the presentation of the cell cycle regulation. Thank you.